Okay, our third speaker is Aaron DiAntonio. Aaron comes to us from WashU St. Louis, whereas he is the Alan and Edith Wolf Professor of Developmental Biology. Thanks so much.
Thank you. All right, well, thank you for the invitation to come talk. I was asked to not give my normal talk about, what we're up to right now in the lab, but instead to give you guys some insight of our journey of going from making a discovery in an academic lab to starting a small biotech to eventually selling that to big pharma and having that drug move into the clinic. And all this work was a close collaboration with my colleague, Jeff Milbrand at WashU. We still work closely together. And so whenever I say I, it's we and obviously great work from the lab. So the last two talks and many other talks we've heard at this meeting focus on one great way to approach CMT and other neurodegenerative diseases, which is to understand the underlying genetic cause and to directly treat the reason the person has that disease. There's a second approach, and that's the approach we take, which is to ask for some sort of commonality in the pathological process of many different neurodegenerative diseases. In our case, it's axon degeneration. So axons are lost, obviously, in CMTs, particularly CMT2s, but of course it happens in one as well as Parkinson's, ALS, glaucoma. Axon loss is an early and often causative event. So we started this project by wanting to understand what are the molecular mechanisms of axon degeneration. Are there any key molecules that are required for an injured axon to degenerate? But we did not know what those molecules might be or what the mechanism was by which axons degenerated. And so as a geneticist, the way to go about trying to learn about a pathway where there's really next to nothing known is to do a genetic screen. And so our logic here was we could grow sensory neurons from a mouse in a well of a dish, and then they grow out axons. And if we cut those axons with a scalpel, the cut axons will degenerate. And so 4 very hardworking young people with good eyes generated about 100,000 individual wells, hand cut the axons in every single one. And in every well, we had a different shRNA knockdown targeting a gene in the genome, just mindlessly every single one. And after a couple years of work, we only had a single gene that was real, and that was SARM1. So when we knocked down or eventually knocked out SARM1 and we injured axons, instead of them fragmenting and degenerating, they persisted beautifully. We were super lucky. SARM1 knockout animals, you can have an animal that has no SARM1, it's alive and well. Other people had made those mice, and they really didn't have much of a phenotype. They live a normal lifespan. But when we injured their peripheral nerves by severing it, where a wild-type mouse ends up with terrible Wallerian degeneration, all the axons are destroyed within a few days. This shows it at a week. These animals, the axons beautifully persist. We did this huge screen in primary mouse neurons. Mark Freeman, who was then at UMass Worcester, had like 5 people work for 5 years to do a conceptually similar screen in Drosophila, and they got 1 hit from all that effort, and it was Drosophila SARM1. So it really suggested that SARM is really central to this process of axon degeneration. Now, it's cool, as a scientist to be able to keep a severed axon from degenerating, but as a patient or a doctor, not too useful, right? The severed axon is not attached to anything. Doesn't matter if it's there or not. It's not going to contribute to a circuit. So we wanted to ask, having found a role in this pathological axon degeneration, are there diseases or situations where axons are lost that are more clinically relevant, where SARM might play a role? And so what we decided to try first, and just as one thing I meant to say was, what I want to give you is what was the data we had generated in the lab that convinced us we should start a company, and then more importantly, convinced people who had money that we should start a company. And so everything I'm going to tell you about is kind of the data we had up until that point. So data piece number 1, SARM's required for axon degeneration. Number 2, knockout mice are healthy, suggesting that if you knew how to inhibit it, maybe it would be safe. Now we need data point number 3, something more relevant than axotomy. And so we chose chemotherapy-induced peripheral neuropathy. So the major dose-limiting side effect for many chemotherapeutics is peripheral neuropathy. And so it's often persistent long after the chemotherapy has stopped, has a huge impact on quality of life, but also has an impact on the efficacy of the chemo, because if it's dose-limiting, by definition, that means many patients can't get their full dose of chemo and don't get a full treatment. So what we found was that in SARM knockout animals, we treated them with vincristine, a commonly given chemotherapeutic. Wild-type mice showed axon degeneration. That was blocked in the SARM knockout animals. They developed pain, so they're much more sensitive. That was blocked. Basically, everything we could look at was completely blocked. So if we had SARM inhibited because it was deleted, you just didn't get chemotherapy-induced peripheral neuropathy. And for chemotherapy peripheral neuropathy, it's okay to treat beforehand because you know when the patients are going to first get their chemo. It's going to last for a certain period of time. It's a well-defined kind of how long it tends to take to cause the disease. And so we thought, maybe somewhat naively, this would be a great first clinical trial. Now, we got a lot of pushback later on because of the oncologists are obviously nervous about doing anything that could impact disease progression. But this is how we were thinking at the time. This would be a great first clinical trial. So now we're really convinced SARM is a great target. We need to understand how SARM works. And nobody knew how it worked. What we did all know was that it looked like a protein involved in innate immunity. And that's because it has something called the TIR domain. And TIR domains are the signature signaling domains for many different innate immune proteins, things like toll-like receptors. And in innate immunity, Nobel Prizes were won describing these proteins where these TIR domains come together, act as scaffolding proteins, recruit signaling molecules that then turn on, cytokine expression, for example. So the model in the field was SARM must somehow be acting as a scaffold to do something. We were able to show, a really great student, Josiah Gertz, showed that the SARM TIR domains, if you could force dimerize them together, that was sufficient to trigger axon degeneration. So it's not just necessary, it's sufficient. And because he now had a mechanism to rapidly turn SARM on, he realized within minutes it led to the dramatic loss of NAD. NAD, that essential cofactor in hundreds of different biochemical reactions. And so we thought this is probably how it's triggering axon degeneration. So normally, in a healthy neuron, SARM is off. Injury somehow activates SARM. We now understand that, and that's opened up new opportunities for treatments. Once SARM is activated, the TIR domains come together, somehow trigger NAD loss, and that gives you axon degeneration. So knowing it's a scaffolding molecule, at the time in particular, it seemed impossible to drug it. That would require some kind of protein-protein interaction, disruption, and although there are tools emerging for that now, it's not a great option. So what we thought was, we need to identify the actual enzyme causing that NAD drop. So we did all the things you'd expect. We thought it was a scaffolding molecule. We did all the co-IPs, we did a mass spec. We tested every known NAD cleaving enzyme, things like PARPs and sirtuins. Nothing worked until a new student came into the lab, who decided he was going to go back to first principles and just purify the enzyme activity, good old-fashioned biochemistry. And what found literally within just like 3 months of joining the lab was probably what will turn out to be the most significant, result in our careers, which is that SARM itself was the enzyme. So we could just purify just the TIR domain of SARM, or eventually in a completely cell-free extract, just synthesize only SARM's TIR domain. And it was a very potent enzyme that could take NAD and cleave it into nicotinamide and CADPR. So this was the moment when Jeff and I were convinced we need to start a company. It's an enzyme. People know how to inhibit enzymes, particularly an enzyme that cleaves a small molecule. There ought to be a small molecule that could, stick in the active site and block it. So we were convinced at this point, but we knew we still didn't have enough data, or we figured we didn't have enough data to actually convince anyone else yet. So the next thing we did was try to prove this activity matters. So we identified the catalytic residue, one single amino acid that is required for SARM to be able to cleave NAD. And that let us then do this functional experiment. So normally, if you're a SARM knockout neuron and you're severed, your axons look fine. They don't degenerate. That's the whole SARM knockout phenotype. If we add back wild type SARM and we sever the axon, well, SARM's job is to trigger axon degeneration. So there it is. Now we add back a SARM missing just the tiny change in the one amino acid needed for this enzymatic activity. And even though you have SARM there, a non-enzymatic SARM does not give you axon degeneration. So that was really a strong demonstration that this activity was going to matter, and really implied that if we had a pharmacological inhibitor of this enzyme activity, we should get the same kind of protection. Now, in general, drug screens are not a great thing for academic labs to do, but we thought this would be our strongest evidence to, convince people with money that we should start a company. And so we did a small scale drug screen in the lab. I think we got really lucky. From just a few thousand drugs, we actually found 2 that were good biochemical inhibitors of SARM1. One actually had a really, IC50 of 150 nanomolar, which is pretty low for such a tiny little screen. And then the key experiment was, okay, do these drugs that block SARM's biochemical activity, if we put them on neurons and injure them, do they protect? And it did protect. So this was really proof of concept. You can find a drug. Now, if any of you, and I'm certainly not, know anything about drug structures, our drugs were terrible. Nobody would put these anywhere near a person. So we didn't have the drug, but we had proof you can find a drug. And if you can find the drug, it could end up doing what we hope. So this is the point where we started the roadshow, and we went out and we talked to VCs, and we tried to convince them, let's start a company to get really good drugs. I'd say the last big challenge is we're academics. We had an MD PhD student who'd made this great discovery. We were trying to publish as fast as possible. The moment we publish, everyone in the world knows it's an enzyme. There's not really any IP here because an idea can't be patented. Yeah, we had a couple of drugs, but we knew they were terrible. We had methods for how to find drugs, but everyone told me, oh, people just get around those patents. So all we really had was speed. And so luckily, we did find partners who were eager to, jump in. That was Atlas Ventures right here in Cambridge. And they assembled a really terrific team. This is a group that works with academics where they just create a company, and they bring in top people from the business end, from the science end, from the clinical end. And it was really, we both learned a lot, Jeff and I, of kind of getting to work with them. So it was all done through contract research organization. So, world headquarters was right here in Cambridge, where there were, 5 people sitting in a little room, basically coordinating all this activity. Those of you in, biotech know all about this, but it was new to me as an academic. We had, in England, we did high throughput drug screening. Was amazing to me. We had struggled for months to get through 2,000 compounds. They did 250,000 in 6 weeks on, with replicates, and just it was really a thing of beauty to see their data. Germany, there were animal models to test in vivo efficacy. Metabolomics was in Spain. We synthesized compounds in India. We did sort of ADME PK and more chemistry in China. So it was this whole kind of world effort. And then it was still really fun as an academic, because every other Thursday morning, Jeff and I would get on the phone with the folks at Disarm, and we'd spend a few hours basically having a lab meeting. We'd hear about the latest data, we'd give suggestions. And so it was exhilarating, and I definitely learned a ton. So then, we came up with pretty good drugs. Here's an example of a Disarm drug. They worked in vivo as well. And so ultimately, it was time to raise some serious money to try to move into the clinic. This was February of 2020. I don't know if you all remember February of 2020. Kind of a tough time to get anyone to do something new and exciting. We were kind of busy. So it then turned out by October of 2020 that we thought the best opportunity was to go with Lilly and to sell the company to Eli Lilly. And then that was 5 years ago now, and this is where we are now. So one thing to know as an academic or, a small biotech that sells to a big company, maybe it's not always like this, but it's like sending your kid to college. They never call, they never text. So I know they're in clinical trials. I hope things are going well. there are many other companies who have jumped on this now. It didn't happen as quickly as I'd thought, but I now know of at least 9 different companies. There are probably more that have SARM inhibitors. There are at least 2 companies that are in the clinic. I obviously have high hopes that that will work. In my last 20 seconds, I'll just say what we've done since then have been a number of things, but one has been to try to convince these companies that CMT2A would be a great model. We know SARM gets turned on by damaged mitochondria. CMT2A is caused by mutations in mitofusin 2, which is required for mitochondria to fuse. And so I'll just briefly show you some data we generated in the rat model that Steve shared with you yesterday. So there's bad axon degeneration in the CMT2 rat. We made a SARM knockout rat. We combined them, completely fixed the axon degeneration. Synapses look really sad. They keep degenerating, regenerating of really atrophic neuromuscular junctions. This is all beautifully fixed. After Mary's talk this morning, I wish I had shown you the muscles. It's really bad muscle atrophy in the CM2 model. Muscles look beautiful in every way we can measure when SARM's also missing. And this shows a functional readout of hind limb grip strength. So someone asked yesterday about SARM, like, why does it work sometimes and not other times to get rid of SARM? And so here we were kind of thinking, even if SARM keeps the axons from degenerating, mitofusin's still missing. The mitochondria are still really sick. It might not help the rat at all, but it did help. And I think part of that reason was that, to our surprise, the mitochondria were actually much healthier. They were still small and round because what mitofusins needed to do is fuse it and give you a big elongated mitochondria. That still happened. But the cristae were better. Localization down to the synapse was dramatically better. There's a big problem with transport in these mutants. So even though mitofusin was still missing, so there's cristae density, which is where the business end of mitochondria are, that was fixed. Axon transport of mitochondria was fixed. So very reasonably, people who'd previously studied mitofusin said it's a mitochondrial protein. We have a lot of mitochondrial defects. It must be because mitofusin's missing. I think the real answer was there's some mitochondrial damage that turns on SARM. SARM cleaves some NAD. You now have a metabolic stress. Mitochondria gets sicker, and there's this positive feedback loop. And so in this case, and there's beautiful work from Tom Schwartz at Harvard showing a very similar result with OPA1 mutations, which causes a different neuropathy. That's another protein involved in mitochondria. And SARM knockout protects beautifully. So in this case, a lot of what had been attributed to the gene knockout is actually secondary to SARM being disrupted. And so I think these are some of the best bets, ones that are mitochondrial or axon transport defects where SARM knockout might help. So last slide, we've done some more stuff to kind of understand the pathway. I told you about SARM enzyme inhibitors. We also identified an allosteric binding site, and there are a lot of companies going after that as a target. We developed an awesome gene therapy. I don't know if Brian's still here, but I think this would work really great for CMT2A if anyone wanted to try it. And yeah, we're just really hopeful that some of these inhibitors to SARM will end up helping people with neurodegeneration. So thank you all for your attention.
Hi, first over here. Yes, online. So I have 2 questions. What's the function of SARM1 outside of neurons? Is it involved in the immune response? And you say that the mouse models don't have any phenotypes, but have you tried stressing them, see how their immune response is?
Yeah, great question. And I always get, why in the world do we have SARM if it's so good to get rid of it? So the immunologists first made the mutant, and because it looked like it would be an immune protein, and they found some modest phenotypes, nothing they were too excited about. There is some data that there's some rare neuronal viruses that probably activate SARM, and then SARM kills that one infected neuron so the virus can't spread. So my biggest concern for a side effect of a SARM inhibition will be an increased likelihood of infections in the nervous system, which like people with MS who are on immunomodulators that happen. So I think that is the most likely endogenous function. But yeah, it's really hard in the mice to find much of a phenotype.
I actually had 2 questions. One was that question, but I want to just pursue that a little bit more, because what about flies? So you should be able to look in the fly then and see, okay, if a virus infects a neuron somewhere.
Yeah, for some reason in flies, it is an essential gene, and flies are dead without SARM, and it is not really clear why. Mice, rats, no problem at all. So yeah, flies have been less useful for that.
Interesting. The second question is, you have this metabolic crisis as the line between SARM and axon degeneration. Any idea what's going on there?
Yeah, I don't, it might be the slide after my acknowledgement slide, if that can be shown. So we do have a pretty good idea of kind of cell biologically what is happening. So we know that first NAD is lost, and then ATP is soon thereafter lost. And that's, you need NAD for both glycolysis and oxidative phosphorylation. Then, yeah, here we go. So we did a single axon analysis and only by doing single axon work could we tell that there was an absolutely stereotyped process of axon degeneration. So what you get is mitochondria stop moving after ATP is lost, then they depolarize, then calcium rushes in, then phosphatidylserine, which should be on the inner leaflet, is exposed on the outer leaflet, and then after that, there's actual breakdown of the membrane.
And is calpain then activated?
Very late. So there used to be excitement about calpain because after calcium comes in, calpain is a calcium-dependent protease. It'll chop up neurofilaments. I think this is after the point of no return. So when we manipulate calcium, structurally, you can take a picture of something that still looks like an axon, but if you look, it's a zombie axon. All its mitochondria are depolarized, they're not moving. It's not a metabolically healthy axon. So yeah.
Thank you for a lovely talk. Just thinking about the other forms of CMT models that it may not work, and of course we don't know, and then thinking of chemotherapy-induced neuropathy, diabetic neuropathy, there's been a lot of excitement about it in many different ones. Do you think it's critical to have a mitochondrial component in the pathogenesis? Do you think that is what is critical, and that there's other forms of axonal degeneration that are accounting for other axonal degeneration?
So without a doubt, there are going to be other things that can cause axon degeneration. But I don't think it'll only be the mitochondrial one. So I didn't get into it, but we really understand how SARM is activated in gory molecular detail. And the most important thing to keeping SARM off is having healthy axon transport. You need there's a negative regulator of SARM that's always being delivered to the axon, has a super short half-life. And so any perturbation such that it isn't transported as well, SARM's going to get activated. And we think this explains the Dunning-Back nature, because if you think about a protein always being turned over, always being transported, where's the point where its levels are lowest? The tips of your toes, the tips of your fingers. That's where you're getting local SARM activation. So if I had to just sort of pick a good guess, I'd say if there's an axon transport defect or a mitochondrial defect, those are your best bets.
And can I just one quick question? Do you think, so I've looked very, very hard in thousands of patients for SARM1 mutations of any type and NMA2. And as you know, there's only about 3 families with NMA2 and none with SARM. Do you think it just could not tolerate gain of function? Do you think that it would just be lethal? And loss of function is very difficult to see because that might be normal people that live to a good old age.
Yeah, so I mean, that is a great hypothesis. So we actually did a study. So, one of the complaints we got when we were trying to raise money was, well, SARM's so important, where's the human genetic evidence that it matters? This is a variant of the question. So what we did is we reasoned, because SARM is normally autoinhibited, there are going to be, as was just described, some loss of function mutants, some neutral mutants, but there'll also be mutants that break that autoinhibition and give you constitutive activity. So there's a very nice collection of ALS patients and controls that have had whole genome sequencing. So we looked at the genomes, and we identified every rare variant in SARM1, every point mutant or small patient. And we did enzymology on every single one. And there were a number of those, about a third of those ended up being gain of function. And then if we asked, are the gain of function ones enriched? They were clearly enriched in ALS, where just overall, they're not. So no, I don't think being a heterozygote for a constitutively active SARM just kills you. We've made mice, they're definitely not dead. But I think that probably does predispose you to degenerative events like ALS.
Last question.
the previous question kind of addressed this already. What, other than ALS, any additional GWAS study where you can maybe, because I know this was always an issue, right? So anything else here in the meantime coming up?
Yeah, I mean, so there is a GWAS hit right near SARM and ALS. It's not completely clear what that does to SARM and if that's really the cause. These rare gain-of-function mutations, Michael Coleman in Cambridge also studied them, and he identified some in HSP patients. So that's a very logical other sort of place where, that it could be occurring. I would, however, say, these were all super rare. So I think they give us a clue as to where SARM could work, but we really understand how SARM gets turned on with these problems with mitochondria and axon transport. So if it's going to have a big impact, it's going to need to be in diseases where there's an underlying pathology that is kind of turning on SARM and not necessarily in the genetic ones.
Okay, thank you. What a great session. Thanks, Will.
So Anna, do we have a break now and then
We-
back at 2:30?
That's correct.
Thanks so much.
See you at 2:30.